Students, I am Dr. Kumar, Assistant Professor of Zoology, Government First Grade College, Shira. In this session is the continuation of the previous class, that is again the phylum Protozoa. In the previous class, we have discussed the locomotory organelles in Protozoa, includes pseudopodia, cilia, and also flagella. Now, again we can see the movement in this session. We will study the movement of an organism using these locomotory organelles. Locomotion in protozoa, that is amoeboid movement, flagellar movement, and ciliary movement. Using these locomotory organs, the protozoans can able to move from one place to another place. So let us study in detail how the animals will move from one place to another place by using these locomotory organelles. The learning objective here, the students can able to understand how the protozoans in protozoans the locomotion occurs using the locomotory organelles. The content of the topic goes like this: is introduction and locomotion in protozoa, that includes three methods of locomotion, that is amoeboid movement, flagellar movement, and ciliary movement. Introduction. As all of you know that locomotion is the movement or the progression through the medium. Animals change their position in search of food, protection, or in search of partners and suitable environment in response to various environmental stimuli. The parts of the cell body performing the locomotion are termed as motile or locomotory organelles. Locomotory organelles in protozoa includes the pseudopodia, flagella, and cilia. Protozoans perform locomotion in three methods, each with a particular type of locomotory organelles. It is the characteristic of the different classes of phylum Protozoa. They are amoeboid movement, flagellar movement, and ciliary movement. The first one is the amoeboid movement, so that takes place with the help of pseudopodia as locomotory organelles. So this can be seen in amoeba. So here it is the amoeboid movement is the characteristic of all the members belonging to the class Sarcodina and certain members of Masticophora and so Sporozoa as well. The amoeboid movement is well exhibited by amoeba. It is carried out by a temporary flowing of cytoplasmic process called as pseudopodia. It moves by extending out pseudopodia and flowing the cytoplasmic content into the pseudopodia. Here you can see these are the cytoplasmic extensions where you can see the pseudopodia. The organism continuously changes its position by putting forth new pseudopodia, while the older ones are drawn inside. So as I told you, amoeba while moving from one place to another place, it extends its body like a finger-like projections. Once it extends its body, so the endoplasm will flow into this finger-like projections, so that the body moves forwards. The formation of pseudopodia depends on the active contraction of the ectoplasm at the posterior end and the forcing of endoplasm into this pseudopodia, so that it will be helpful in propelling the body forwards. You can see the pseudopodias are here, and the endoplasm, whatever the cytoplasm which is present inside the body, is called as endoplasm. Once the pseudopodia is formed, the endoplasm will flow into this finger-like projection, so that the body can be able to move forwards. The next is flagellar movement. It is the characteristic feature of the members belonging to the class Mastigophora, which bear one or more flagellas. It involves active swimming in a liquid medium. Flagella is a fine, delicate, thread-like extension of the protoplasm. There are three types of flagellar movements have been recognized, that is undulating movement, simple conical gyration and paddle stroke movement. Let us go one by one. First one is the paddle stroke movement. So in this movement of here the flagellum is sideways lashes consisting of an effective stroke and a relaxed recovery stroke, in which flagellum strongly curved and brought about the forward again. As a result, the animal move forwards. Here in this slide you can see the flagellum. So there are two movements. One is the effective stroke and the recovery stroke. In the effective stroke, the flagellum it will starts beating backwards in an effective manner, so that body move forwards. So in a recovery stroke, so this whatever the flagellum which is bent, so it will become straightened. So in this recovery stroke, usually there is no significant movement occurs. Undulating movement. So it is a wave-like undulations in flagellum. So during movement, during effective stroke and recovery stroke, so usually the flagellum moves. When these undulations proceed from tip to the base, that means the tip to the base. When the movement that occurs from tip to the base, so it will helpful in the movement of animal forwards. And at the same time, the backward movement is possible when this undulation passes from base to the tip. When this undulation passes from base to the tip, backward movement occurs, and when this undulation passes from tip to the base, forward movement occurs. Okay. So when such undulations are spiral in nature, once they occurs in spiral manner, that causes the organism to rotate in a spiral manner. The next one, last one, is the simple conical gyration. So gyration here is the movement of animal in a spiral manner along the movement of the axis, the movement axis. Okay. So it is a spiral turning of flagellum like a screw. That is the spiral movement of flagellum like a screw. So this exerts or propelling action, pulling the animal body forwards in a water medium. Okay. The rotation is what is called as gyration. The meaning of the gyration is the revolving in circle. The animal, so they starts moving in a circle along with the movement axis. Okay, around the axis of movement. The energy it is utilized for this gyration movement is provided in the form of ATP. The next third one is the ciliary movement. As all of you know that the cilia are the small hair-like structures which are very much present on the surface of the body, throughout the body. Ciliary movement is the characteristic feature of the class Ciliata. Ciliates are the fastest of all protozoans. They swim at a rate of 400 to 2000 microns in a second. It involves the active swimming in a liquid medium. It is brought about by the permanent hair-like structures called as the cilia. The ciliary actions resemble the swing of a pendulum. Here you can see like flagellum here in ciliary movement also you can see both effective stroke and recovery stroke. Effective stroke here it is called as power stroke and recovery stroke. In this movement of cilia, the cilia will bend backwards. The bending of cilia backwards is what is called as effective stroke, and at the same time they become straightened and they are recovered. That is what is called as the recovery stroke. So that is the bending and straightening of the cilia that occurs alternately. Okay. The backward effective stroke is more effective during the movement. The effective stroke always brought the body forwards in the liquid medium, and the recovery stroke has no significant movement. The water moves in the direction of beat. The water moves or the animal moves in the direction of beat, while the ciliates tend to move in an opposite direction. Once the beating occurs, the body propel forwards in the water medium. The cilia present all over the body do not pulsate simultaneously. So not all the cilia will beat simultaneously. This is the characteristic feature of ciliates. In fact, the wavy pulsation passes from one end of the body to the another end of the body. And this beating or pulsating of the cilia, so that starts from one point and ends in the same point. Okay, there is no simultaneous beating of the cilia occurs. The same thing again, the effective stroke and the recovery stroke. During effective stroke, the cilia will bend backward so that the body move forwards. At the same time, the cilia becomes the straighten and becomes the normal position, what is called as recovery stroke. Okay, so the wavy pulsation that passes from one end of the body to the other end of the body. Okay, so dear student, this is about the locomotion in protozoa. So that is carried out by three different methods using three different locomotory organelles, that is pseudopodia, amoeboid movement, the flagellum, that is flagellary movement, and cilia, ciliary movement. Thank you.